The overall goal of this miniaturized glycan array is to analyze the specificity and avidity of Influenza A virus hemagglutinin.
This assay can help to answer key questions regarding Influenza A virus receptor binding and avidity. The main advantage to this technique is that within a single assay we can address specificity as well as relative binding avidity, normally not achieved within typical glycan array and plate-based assays.
This protocol begins with preparations of the glycan samples. The base solution for the glycans is a stock printing buffer, which, when made, should be slowly titrated to a pH of 8.5, and then given an addition of surfactant. Using the prepared printing buffer, dilute the PAA conjugated glycans to 100 micrograms per milliliter. Then, dilute the monovalent glycans to 100 micromolar, also in the printing buffer. Finally, make working stocks of the amine functionalized dyes at 1 micromolar. Now, load 10 microliters of each glycan sample into a well of a 384 well microtiter plate. This is enough solution to print 5 slides. Also transfer 10 microliters of the dye marker to 1 well of the printing plate. Prepare to print